Okay, we are in the meat of cell respiration right now, and we're going to finish this up. This is the more difficult part of this chapter. Okay, so I've already introduced you to cell respiration, what it is, parts of it. The point is, of course, to oxidize, strip off hydrogens and electrons from a glucose, and in the process of oxidizing it all the way to carbon dioxide, we are going to reduce oxygen. We're going to make some CO2, obviously, from the oxidation of the glucose. We're going to make some water from the reduction of the oxygen, and the aim is to make as much ATP as possible. That's the summary of cell respiration. Okay, first, glycolysis, breakdown of glucose, glycolysis. All right. There are different pathways for glycolysis. We are going to only look at the major one, the Emmon-Meyerhof pathway. Two guys, Meyerhof and Emmon, worked it out. At the bottom here, you'll see reference to the other ones, the Ender-Deuteroff pathway and the pentose phosphate pathway. There are different ways to oxidize glucose, producing different intermediate byproducts in the production of reduced electron carriers and a little bit of ATP. We're going to look at the major pathway. All three of them are glycolytic pathways because they're all ways of breaking down glucose. All right, I've already introduced you to substrate level phosphorylation. I'll show you that. And we're going to start with glucose. Okay, so in this process, on the picture you see here, whoops, you see glucose. Now, in lecture, I'm going to show you the individual chemical reactions, but what you see right here, there are about 9 or 10 of them, is the sum total of the glycolytic pathway. Your starting material is glucose. That's what you got over there. Here's the input of a couple of ATP, and we're going to input a couple of potential electron carriers, NAD or NAD+. It's oxidized, remember. That's why it's called NAD. It is not reduced. It doesn't have any electrons. All right. Basically what we're going to do, and again I can show you this much better on an animation in class, but to summarize this, we're going to add phosphates onto the glucose. That's why we need 2 ATPs. Remember adenosine triphosphate, and remember that adenosine triphosphate has high energy bonds with a phosphate. All right, so we're going to split off that high energy bond, and we're going to take that phosphate with this high energy bond and we're going to stick it onto a carbon of glucose. And we're going to do the same thing with a second ATP. We're going to take another high energy phosphate and we're going to stick it onto another carbon, and we've now made a diphosphate molecule. It's now called fructose diphosphate. We have stuck a high energy phosphate on both ends. The next thing we're going to do is we're going to split that molecule into 2, making a 3-carbon molecule. Glucose has got 6 carbons, and actually I should have split it that way. No, I'm not a very good drawer, am I? Okay, so we're going to actually, I'm going to use a dotted line, I think. There we go. I'm going to split it that way, and we end up with 2 3-carbon molecules. Each of these 3-carbon molecules will have a high energy phosphate bond on them. We are then going to knock off that high energy phosphate bond. We're going to add it to ADP, and we're going to make ATP. We're going to do that a second time. We're going to make a couple of more ATP. Notice we had to input 2 to phosphorylate to begin with, but we get 4 at the end. Now, along the way, during the manipulation of these two little 3-carbon molecules, there's going to be an oxidation reduction reaction, and we are going to give a couple of electrons to NAD, and it becomes reduced to NADH. Now, at the end of all of this, what has happened to glucose right there? Well, we've ended up with 2 of these 3-carbon molecules. These are pyruvic acids. You see 2 right there. I'm going to switch over to red. Okay, so we have 2 molecules. It's a small 3-carbon molecule. There's really not much else to say. Down here is substrate level phosphorylation, which happens during the glycolytic pathway. And again, what I said before, and I just said a minute ago, that high energy bond right there and that phosphate right there, we're going to knock that off, stick it onto ADP, make an ATP. That's how we're making those 4 ATPs. There's no electron transport chain, anything like that as part of this. Now these NADHs, I haven't told you what we're going to do with them. They are obviously, because you should remember this, are going to be involved in the electron transport chain at the end of all this. Okay, so that is glycolysis in a nutshell. Notice I didn't say the word oxygen anywhere in there. No oxygen involved. Now remember, that's happening in the cytoplasm. Alright, so now, before we go into the Krebs cycle, we need to convert pyruvic acid into a different molecule, because pyruvic acid is not involved in the Krebs cycle. This is sometimes called the intermediate step or the transition step from glycolysis to the Krebs cycle, because it's acetyl coenzyme A that actually is involved in the Krebs cycle. It's actually the first reactant of the Krebs cycle. This is just a one-step little chemical reaction. And again, in eukaryotic cells, this is where we would transition into the mitochondrion, but in bacteria, there is no mitochondrion involved, and we move to the membrane, ultimately, cell membrane. Okay, so you'll notice a couple things here. Number one, here is our electron carrier. We're going to have an oxidation reduction reaction. There's going to be a pass-off of electrons. Notice this guy right here. Notice there's a hydrogen here. We're going to give electrons and hydrogen. That's going to end up reducing NAD. There's our NADH right there. And what do you got left? Look at this guy right here. Remember, we're going to break that bond right there. Look at your CO2 right there. Now, obviously, this is going to chop off that side chain, and what we have left is that little CH3 and a double bonded oxygen with a carbon. But notice on our acetyl coenzyme A, we've tacked on another side chain. So there's our acetyl coenzyme A. Okay. Now, one last point. Remember, in glycolysis, we split a glucose into 2 pyruvic acids. So we're really going to do this twice. We're going to do this to each pyruvic acid. We end up with 2 acetyl coenzyme A's. Okay, so here's the Krebs cycle. We're going to start with acetyl coenzyme A. Remember that this is a cyclical pathway. So we've got about 12 chemical reactions here. You start out with a reactant. We end up with end products. Those end products become the reactants for the next chemical reaction. Their end products end up being reactants for the next chemical reaction. And the last, let's say that acetyl coenzyme A starts into the reaction right there. The last thing made in the very last reaction is oxaloacetic acid right there. Those two things are going to react together. There's the first reaction of the Krebs cycle, named for Hans Krebs, also called the citric acid cycle, because when those two things come together, you get citric acid. In this process, let's see, let me go to the next picture. I'll get back to this in a second. Here is the easy reaction to look at. It's a little bit off of the screen, unfortunately. Oop. I can't make it smaller. Okay, so here is our acetyl coenzyme A. Let me get rid of the bar. Okay, here's our acetyl coenzyme A. Remember, we're going to have two of these. It reacts with the last thing in the cycle, oxaloacetic acid. I'm just going to call it OAA. These two together form citric acid. There's your chemical reaction number 1. Notice that there's a 1 right there. Okay, so there's our citric acid. Now we're just going to kind of tweak the molecule. We're going to kind of change it. Notice that it's a 6-carbon molecule. Acetyl coenzyme A had 2 carbons, and oxaloacetic acid had 4 carbons, so that's how we get a 6-carbon. Okay, so anyway, we tweak the molecule. We now have isocitric acid. Isocitric acid, we are going to pass electrons and hydrogen from isocitric acid to our electron carrier, NAD. NAD becomes reduced to NADH. Isocitric acid is going to become oxidized to ketoglutaric acid, but there's one other little thing we're going to do. We are going to knock off another COOH. And notice that we are making a CO2. That's where the COO goes. Hydrogen kind of wanders around. So there's our ketoglutaric acid from that reaction. Okay, ketoglutaric acid, we're going to pass off a couple of more electrons to NAD. Ketoglutaric acid is getting oxidized to succinyl-CoA. We're going to knock off another carboxylic acid group. Look at that guy right there. There's our CO2. That's where it goes. We're going to tack on a coenzyme A on the molecule. That's how we make succinyl coenzyme A. Now, succinyl coenzyme A, we are going to knock off the CoA. There you see it's going off. We're going to add a water molecule. We are going to make a high-energy molecule by substrate level phosphorylation, because that's a high-energy bond right there. We're going to pass that high-energy bond over to GDP instead of ADP. We're going to make GTP, which is the cousin. Whoa, that's weird. Uh-oh. Trying to get rid of my marks. There we go. Okay, so... all right. Where was I? Oh. So we have made GTP, and that GTP then is just changed over into ATP. It's a close cousin. Now we've made succinic acid. Succinic acid is going to donate electrons to FAD. FAD is a lower redox carrier, less energy. It carries electrons at less energy than NAD does. It gets reduced to FADH. Succinic acid gets oxidized to fumaric acid. Fumaric acid gets tweaked to malic acid. There's, again, notice we got 4 carbons right there. We're going to pass off another couple of electrons and hydrogen to NAD. Malic acid gets oxidized to oxaloacetic acid. NAD becomes reduced to NADH. And now remember that there were 2 acetyl coenzyme A's. The second acetyl coenzyme A is involved, and the cycle goes a second time. So everything, I'm trying to get rid of, don't know why, but something's not working here. Everything in this cycle is produced twice from one glucose molecule. Okay, so here's just a look. This is a nice picture showing you how we are reducing the carbon number in the molecules. Not important to know, but again, over here is our oxaloacetic acid, or oxaloacetate. Difference is just one hydrogen, doesn't really matter. It's a 4-carbon guy. It reacts with the 2-carbon guy, acetyl coenzyme A, 2 and 4, boom, there's citric acid, there's our 4-carbon. Okay, so what have we done in aerobic respiration? Let me go back to this. All right, we made a lot of NADH, many molecules. We've made a couple of FADHs. We made bunches of CO2. We made a bunch of intermediate organic compounds like ketoglutaric acid, succinic acid, and malic acid. And many of these things are used for other things that the cell needs. We made a little bit of ATP by substrate level phosphorylation, and there we go. So let me go back to the next one. Okay, so here again is the very last step. Okay, so the last part, electron transport chain, you see the main components here. We're going to start with the electron load that's being brought in by NADH, some of it brought in by FADH. Remember, we made most of this from the TCA cycle, citric acid cycle, Krebs cycle. We also made a little bit from glycolysis, remember, also. I should have also put glycolysis here, because we made a couple in glycolysis. Can't write very well with this. Okay. Now, the other electron carriers of the electron transport chain, or the electron transport system, flavomononucleotide, FMN, a variation of riboflavin, and FAD is flavin adenine dinucleotide. It's also a variation of riboflavin, and NAD is nicotinic adenine dinucleotide, which is also a variation of nicotinic acid, another one of the variations of the B vitamins. Okay, we also have quinone, also called coenzyme Q. And then we have the cytochromes B, cytochrome C, cytochrome A. These are iron-based organic compounds. And then finally, we'll talk about oxygen. Okay, so let me show you a picture of it, and then we'll get back to some of the other things on here. All right, so here's a look. Yep, it's off my screen. Let me see if I can make this bigger. Put it up higher so it can be seen. There, now you can see the bottom. All right, so let me point out where you are. You are looking at, in a bacterium, this is the cell membrane, not cell wall. Forget cell wall. The cell wall is actually out here. Cell wall. All right? And this is the space right there between cell membrane, cell wall. Cytoplasm is down in here. All right. Now remember, if this is eukaryotic, you would be looking at a membrane inside of a mitochondrion. But, all right, so follow me through here. Embedded in this membrane are electron carriers. Okay, so here is the entry of NAD, H with its electron load. The electrons are passed to FMN, and FMN, it's not shown here on this particular picture, but it's going to be part of this guy right here. FMN becomes reduced in NAD. H becomes oxidized to NAD. And that then can go back and pick up a couple more electrons in the Krebs cycle. All right, FMN is going to pass these electrons off to coenzyme Q. I'm going to put it right there. So ubiquinone or coenzyme Q, or just called quinone. It becomes reduced and FMN becomes oxidized. Quinone passes them off to cytochrome C, becomes reduced. Then it passes them off to cytochrome B, which becomes reduced. Then ultimately, the electrons get handed off to cytochrome A, which becomes reduced. Now, by this time, more NADH has started passing electrons down the electron transport chain. Now, at the very end of this chain, if you look at the far right, look at oxygen right there. It's the final electron acceptor. When it picks the electrons up, it becomes reduced to water. There you go. Now, let me go back to the previous couple of shots right here. All right. Now, I haven't told you how we're going to make ATP yet, because I don't think I even mentioned ATP in that 5-minute description. We'll talk about that more in just a minute. Now, look in red at this line next to the bottom, one up above the bottom. Aerobic respiration is where the oxygen and oxygen molecule serves as a final electron acceptor. If any other non-oxygen molecule is used, for example, it could be a nitrogen molecule, a compound like nitrate or nitrite, NO3 or NO2. It could be a sulfur compound like sulfate, sulfite. If the organism can use a non-oxygen electron acceptor to pick up those electrons, it's called anaerobic respiration. Everything else is the same. It's the same Krebs cycle. It's the same glycolysis. The only thing different is the very last step. Who's the final electron acceptor? By the way, our cells do not do this. Eukaryotic cells don't do this. A lot of bacteria do. Okay, now we need to talk about where the ATP is coming from, because I haven't mentioned it. All right, so look at this line in the middle. Energy from the electrons pump protons, hydrogen ions, across the membrane to the space between the membrane and the cell wall. And that's going to establish a proton gradient. Okay, so let me take you back to this picture and notice hydrogens. There are references to hydrogens here. Well, remember that this is the space right in here between the cell membrane, I'm going to put CM, and cell wall. Hydrogens get pumped out here. I'm drawing hydrogens in case you haven't noticed that. So that means we have a huge amount of hydrogens between the cell wall and cell membrane, much greater than inside amounts of hydrogen. Let me show you this on a different picture. All right, here's another picture. So in the bacterium that you see over here, again, there's the cell membrane. The cell wall is out here. This is cell wall. So here are the electron carriers. You can see the path of the electrons, carrier to carrier. For example, you can see I highlighted them in yellow. Here's your oxygen right there. There's the water from the reduction. All right, now look at these plus icons. These represent hydrogens. You can see some hydrogens out there. They end up going out into that space. And that creates a huge surplus of A, hydrogens, and B, positive charges. So what we have here is an electrical gradient because there are now more positives out here. Let me put more positives out here, fewer positives inside. And we also have a pH gradient, because remember that hydrogen ion is directly related to acidity. So now we also have a difference in pH potential. Okay, so let me point out this guy right here. And there's a better picture maybe of him right there. This is called ATP synthase, or ATP synthetase, and it's basically a little chemical motor. All right, now look at these hydrogens. There's a hydrogen, the little balls right there. You have a motor. Okay, first of all, you've got a little motor right here, and the hydrogens can go through this. There's a object right over here called a stator that you don't have to know, but it's a non-moving structure holding this first little motor to this second motor down here. The hydrogens are flowing through this. This is a open protein channel embedded in the membrane. You can see that on this picture over here. It's also an enzyme. So as the hydrogens flow through, the energy from the flow of the hydrogens, that energy is converted by this enzyme, ATP synthase, with the addition of ADP. That energy is converted into, or that potential energy of the hydrogen flow is converted into ATP. Now, what is this called? This is called the proton motive force, or chemiosmosis. The Nobel Prize for this was gotten in 1976 by Peter Mitchell, and it is the way ATP is made in cell respiration. So there are differing amounts of energy, ATP, that are made depending on whether the electron donor is NAD or the electron donor is FAD. And that has to do with the number of hydrogens. NADH, because it's at the beginning of the electron transport chain, dumps more hydrogens, causes more hydrogens, let me rephrase that, to end up out here. FADH, when it's the main carrier, it bypasses this first complex right here. And because you're bypassing the first complex, because NAD is not the first carrier when it's FADH, you don't get quite as many hydrogens out here in the space. You get a little bit less ATP made that way. Okay, so how does that translate? Here's a debit credit sheet. Nothing that you need to remember from this, but I was just trying to show you where all the ATPs come from. Okay. Erase that. Okay. So I just mentioned this. For every one NADH that brings in electrons, you get enough hydrogen ion out there in the space to create that hydrogen flow to make 2.5 molecules of ATP. Again, it used to be thought of as 3. And if FADH is the carrier, but it's only the carrier for the electrons one place in the Krebs cycle, you get a little bit less hydrogen ion, and that makes about 1.5 ATP in the proton motive force. Now, if you take all of these, here's everywhere where energy is coming from. From the glycolytic pathway, we made 2 reduced NADHs. Remember, we also made ATP directly by substrate level phosphorylation. We made 2 of those. 2 NADHs translate into 2.5. Wait a minute. For each NADH, 2.5 ATPs, so 2 times 2.5, 5. From the intermediate step, we got 2 times 2.5, 5. We got a couple of, remember GTP, 2 GTPs, and it was turned into ATP? Okay, so substrate level phosphorylation, 2 of those. Then we got 6 NADHs, 2.5 ATP, 15. Then, at one point, the main carrier is FAD. Remember, we only get 1.5 ATP times 1.5. There's 3. You add all that up, and max that you can get is 32. Now, you're used to probably seeing, let me change color here, that this would be, instead of 2.5, it would be times 3. So you would see 6 there. That would be 3. That would be 6 there. And this would be 2. 2 times 2, 4. And down here you would have 3, and that would be 18. So the difference would be 12, 16, 18, 20, 38 ATP. And remember that 2 have to be sacrificed at the beginning of glycolysis. So if you subtract the 2 from 38, you get 36. You're used to seeing 36, but actually they think it's now probably less than 30. Again, that's more info than you need, but just so you understand what's going on. All right, so let's shift gears and go to fermentation. Food production. I showed you lots of foods at the beginning. Here's more. Olives, bread, cheeses, blah, blah, blah. Okay, different ways you can look at fermentation. I'm not going to go over all these. You can look at them. But notice a couple things. First, no oxygen involved here at all. It's the way alcohol is made. Lots of foods are made this way, like olives. But also, this also causes food spoilage as well as food production. Frankly, some people would say that some cheeses like Stilton and Limburger are spoiled cheeses and they wouldn't touch them with a 10-foot pole, whereas other people will pay $12 a pound for a piece of Limburger. So what's spoiled to one person is not necessarily spoiled to somebody else. Anyway, it's inefficient compared to respiration because you're only getting a few ATPs as opposed to, again, 30, 32 from every glucose that you get in aerobic respiration. We like many of these byproducts, so that's why it's useful to us. Okay, there are lots of different fermentation pathways. I only want to show you 2. All right, so first, we start out with glycolysis because it's the common pathway. Common pathway. We end up at the end of glycolysis with pyruvic acid. There's pyruvic acid. That's our starting place for the fermentation metabolic reactions. The first part is glycolysis. Glycolysis is common to both fermentation and cell respiration. All right, now there are 2 pathways I want to show you. One you probably already know, and that is the lactic acid fermentation cycle, pathway. If you've had A&P, you've covered this. So in this fermentation called the lactic acid fermentation, you start with pyruvic acid. Notice it's a 3-carbon molecule. NADH is going to give electrons to pyruvic acid. NADH becomes oxidized to NAD, and pyruvic acid becomes reduced to lactic acid. You're hearing a cat meow. That's my kitten. Notice the differences between pyruvic acid and lactic acid. There's a hydrogen right there where I put a little dot. Again, that's coming from the giveaway from NADH. That's lactic acid. It's a 1-step pathway. That's the lactic acid pathway. There are a lot of foods made with lactic acids. These dill pickles right here, sauerkraut, yogurt. Those are all lactic acid pathways. I mentioned that if you had A&P, you'd know it because when you covered muscle metabolism, you covered lactic acid fermentation. Muscle cells can do this when they're not getting enough oxygen, and that should remind you of muscle fatigue and lactic acid accumulation. All right, and oxygen debt. All right, now the other pathway, let me get rid of that, is the alcohol pathway, which you see going over here to the right. All right, so here's our pyruvic acid. What we're going to do is we're going to knock off a carboxyl group, which is this guy right here, and that's going to make CO2, which you know is a byproduct of beer and wine and things like that. What we have left is acetaldehyde. It then gets electrons from NADH. NADH becomes oxidized to NAD, and acetaldehyde becomes reduced to ethanol. And again, notice that there's hydrogen here that was stuck onto that oxygen. So that's the reduced end product, ethanol. You see the alcohol fermentation over here. You see the lactic acid fermentation over there. Again, we're only making a couple of ATPs. Let me skip over that. All right, so here's a look at some different kinds of routes of fermentation and organisms that do it. I mentioned lactic acid fermentation. So here is our pyruvic acid. Go to the left, you see lactic acid. Again, yogurt, cheeses, streptococcus, and lactobacillus do that. On the other hand, yeast, like Saccharomyces, go the alcohol fermentation, making ethanol. But you also see there's some other fermentations here. For example, Clostridium does a more involved kind of fermentation, which can end up making isopropyl alcohol. One of the organisms we use in lab, Enterobacter, ends up doing a fermentation that makes butanediol. So there are lots of different variations of this. Okay, so here's a look at, again, different routes like lactic acid, dairy products, different acids, vinegar, for example, acetic acid, cheeses, lactic acids, alcohols, not just ethanol and isopropyl alcohol, but methanol, and various chemicals that are used in processing plants and industry and things like that. And then there are a lot of foods that we're not familiar with from other countries that are fermentation products. If you've ever been to Hawaii or you go there, you probably are going to want to try once anyway. I don't know if I try it more than once. Poi, which is taro root that's been fermented by lactic acid bacteria. Most people say it tastes kind of like glue that has a little bit of purple dye that's slightly acidic. You have to grow into it. All right, so here's a look at the three processes that we've covered. Okay, so reminder, we just did fermentation. Notice only a couple of ATPs. Sometimes you can get a little bit more, but not very much. Then we covered aerobic and anaerobic respiration. Remember, the only difference is who's the final electron acceptor. Remember, you have the Krebs cycle, you've got glycolysis, you have an electron transport chain in both processes. Because oxygen is not the final electron acceptor, you don't generally get as much energy in anaerobic respiration as in aerobic. But it's certainly more than fermentation. All right, other pathways, just in passing, because we're going to have this in lab. We've been talking about glucose, but glucose is not the only thing that can be used by microorganisms. We're going to be using some of this media that you see over here listed on the right with these nutrients, we use starch agar. So some bacteria have the ability to make an enzyme, amylase, that breaks starch down. The starch gets broken down to maltose, which gets broken down to glucose, and then we can shunt it into aerobic respiration. Some organisms can use lipids that can be shunted into the Krebs cycle and into glycolysis. Some microorganisms can use protein derivatives and amino acids to make energy. But again, the carbs are generally going to be the ones that are preferred because they're the easiest to break down. And again, here's just a look at the carbohydrate guides that are used for energy in the middle, the glycolytic pathway, lipids over towards the right, and proteins and amino acids being used on the left. But they all, again, come into either glycolysis or the Krebs cycle, and in all cases you can end up with ATP down there at the bottom. Okay, that is it.